This takes forever, this conversion. Let me see whether I can just share instead my screen. Where do I do that? Start sharing. Here.
Yes, we have it.
Does that work for you? Do you all see my screen?
Yes, we have it.
Okay, that's how we'll do it.
Yes, Dr. Do you have anything? Yes, Dr. Do you have anything? Dr. Weigel, can you hear me? Dr. Weigel, can you hear me? Dr. Weigel, can you hear me? Dr. Weigel, can you hear me?
Close my doors.
yes. So I guess
Am I loud enough?
I just want to welcome you and yes.
Good.
So I guess I just want to welcome you again. Thank you for your time in our session. Session. I will start your time and the session. We will start in about 10 minutes.
Mm-hmm.
Thank you so much.
Can you hear me? Okay. So I think we can start.
Can you hear me?
Yes.
So morning everyone.
So I think we can
it's a
start.
Pleasure to introduce.
So morning everyone.
From Max Planck Center
It's
Biology
a
in
pleasure
Tübingen.
to introduce Professor Weigel.
Introducing him, I think it's
from Max Planck Center Biology in Tübingen.
necessarily going to be very brief, given
Introducing
the
him, I think is necessarily going to be very
just for
brief,
people who are not familiar.
given the breadth and depth of his work.
Is the voice
Just for
very
people who are not
reverberated?
familiar, so sorry. Is the voice very reverberated?
Okay. Okay, am I heard? Okay. Yeah, sorry, there was a bit of vibration.
Okay, am I heard?
Yeah, the sound is good. The sound is good.
Okay. Yeah, sorry, there was a
So
bit
Professor
of
Weigel
vibration.
earned his PhD in genetics at Max Planck Institute in 1988.
So Professor Weigel
did a postdoc at
earned his
Caltech
PhD in
between
genetics at Max
1989
Planck
and
Institute at 1988.
and was an assistant
And
professor
did a postdoc at
at
Caltech
Salk
between
Institute in
1989
Lahore,
and 1993, and
between
was an
1993
assistant
and
as
2002,
professor
and he has been the
at Salk
director
Institute at Lahore
at the Max Planck Institute for Biology
between
Tübingen
1993
since
and
2001.
2002. And he has been the director
He's received numerous awards for
at
his
the Max
work,
Planck Institute for Biology
including
Tübingen since 2001.
Wilhelm Leibniz Award.
He's
in
received numerous
2007,
awards for his
Fulbright Award in
work,
2010,
including
the GSA Medal,
Wilhelm Leibniz
the
Award
prestigious Genetics
in
Society of
2007,
America
Otto Award in 2010,
in 2016, and
the
Barbara
GSA Medal,
McClintock
the
Prize
prestigious
in
Genetics
2019.
Society of America
and he's
in
a member of
2016,
numerous
and Barbara
societies
McClintock
and
Prize
many editorial
in
boards.
2019.
I would
And
really
he's
suggest to look
a
at
member
his
of numerous
long CV.
societies and on
His work
many
is
editorial boards.
quite
so
interesting because
I would really
he has
suggest
been
to look at
combining
his long
developmental and
CV.
evolutionary
His work is
questions,
quite interesting because
using
he has been
mainly,
combining
although not exclusively,
developmental and
Arabidopsis
evolutionary questions
and
using
today we have the pleasure of hearing
mainly,
him talking about
although
one
not
of his
exclusively,
latest,
Arabidopsis thaliana.
works. And
Today we have the pleasure
really
of hearing him talking
Student
about one of
Scientific
his latest
Association of Agronomy and Plant
works.
Breeding at my
And,
old alma mater,
I have to really
University
thank
of
the
Tehran,
Student Scientific
to,
Association of
give us this
Agronomy and
opportunity
Plant
to
Breeding
hear,
at my old alma
Professor
mater,
Weigel,
University
talking about
of
this
Tehran,
fascinating
to
question about the nature of mutation.
give us this opportunity to hear Professor
please.
Weigel talking about this fascinating question about the nature of mutations. Professor Weigel, please.
Thank you. Yes, a little bit about myself. As you just heard, I actually got my PhD at the very same institute where I'm now in 1988, and I was working on Drosophila embryo development. As a doctoral researcher, I discovered the Fox class of transcription factors, which I'm sure many of you have heard of. I thought at the time that Drosophila biology might not have a future forever. That was obviously completely mistaken. But I thought I have to do something differently. I got wrong advice that mouse genetics probably wouldn't be feasible for a number of years, and I ended up working on plants, which I have not regretted at all. Initially, it was plant development. Initially, it was on how flowers are patterned, and flower patterning got me to think about the initiation of flowers, flowering time, and that is actually a very important adaptive trait. It's very important when in a season a plant flowers. So at the end of the last century, in the late 1990s, together with my good friend and colleague Joanne Corey at the Salk Institute, started to think about very seriously about genetic variation, and this is what we've worked on since for the last 25 years about. And there are different aspects to our work on genetic variation. Really a chance event led us to think about plant immunity, which I will not talk about today. That's an important part of my lab's work. And the other part is really the variation in genomes, and this is what we'll discuss today. I like to comment on things that are important to science, but also in scientific policy, especially with respect to genome editing. And you can follow what I say there on my Twitter account, Plant Evolution. Before we start, a quick disclaimer. So some of the other organizations from where I have financial interest, in addition to Max Planck, is Howard Hughes, which I advise, KWS, which I advise, and then eLife, where I'm a deputy editor. So I think everybody here subscribes to the idea of Darwin's theory of evolution by means of natural selection. And this is the first phylogenetic tree that was ever drawn. Again, I'm sure you all recognize it, drawn by Darwin himself. And mutations are not the only thing that drives evolution and adaptation, but mutations are, of course, a very important fuel in evolution and adaptation. Without mutations, evolution eventually would run out of material to work on. So why is it important that we understand adaptation? Well, I think in this day and age, this does not really need much explanation. I brought here a figure from a paper that is almost 10 years old now, Nature Climate Change, and it gives here a map of the world, and it's colored by this permadrought severity index, which indicates the change 2100 versus present of the danger or risk of severe to extreme droughts. And what you'll see is that Central Europe here, interestingly, has some of the largest threat. And that's because currently, or until very recently at least, rainfall was always very abundant, and there was essentially not much risk of extended drought. And that will change in the next decades, unless, of course, we take clear action. You'll see here, Iran has somewhat moderate risk of a drought. Depends a little bit in which area. Some of them also here quite high. Now, how do organisms adapt to climate change? The conventional scenario is that organisms adapt by migration. So what you do is you look at where is an organism found today, a species found today, and then you look at where will you have the same environment in some years' time, and then you predict it will just move north if you're in the northern hemisphere, for example. Or if you're on a mountain, typically it will move up a mountain. Now, that has a few problems, not the least that plants are, of course, somewhat limited in the way they can migrate, although, of course, seeds and pollen can travel quite far. But again, so this is the conventional scenario, and this is from an actual paper, almost 20 years old now, predicting the distribution of this specific species here, what it is like in Great Britain today or 20 years ago, what it would look like under moderate climate change or extreme climate change, and you just see this northward movement. And the problem is that this scenario relies disproportionately on population at the very edge. So these populations here have to move in that direction, and these and as well the ones here. So populations at the edge are actually the least adapted and diverse populations. So they are the ones that are actually least in a position to adapt to a new environment. So we're studying this with Arabidopsis thaliana. This is a little bit out of date. It's a map of the world again, and it shows accessions from all over the world And specifically, these are the accessions that we sequenced in the 1001 Genomes Project. And I'm saying it's out of date because we now know, for example, that there are many places in Africa where Arabidopsis thaliana is found, and that Arabidopsis thaliana has a very long history in Africa, much longer than in Europe and Asia. And it has been introduced to North America as well as South America and here Australia in historic times. So it grows in very different places. This is near Tübingen, and then a train station, oak forest here in Spain. This is not a staged picture. This is actually a picture from the Baltic Sea. This is an abandoned cloister here in Ireland. This is in Patagonia in South America. So just looking at this, you can easily guess that this plant had to adapt to very different environments. So Moy Exposito Alonso, when he was in the lab, he is now leading his own group at the Carnegie, a number of years ago, conducted a simple large-scale common garden experiment to test how well adapted different Arabidopsis populations already are to different climates. So he made use of the 1001 Genome Project, about 500 different accessions, and then he grew them in two different places, either here in Tübingen or here in Madrid. In Madrid, there's naturally low precipitation. Tübingen is naturally high precipitation. This is a map of the accessions where they all came from. And he grew them in these rainout shelters so that he could control how much water these plants received. And you can see here right away, if you simulate rain, so if you give quite a bit of water, the plants do well. If you give less water, mild drought, the plants don't do so well. Total, Moy grew 24,000 pots, which he imaged at 20 time points, and then looked at both survival and seed counts. And he did this both for populations, so about 20 plants per pot, and then individual plants here. And so these are the total observations. You're looking at here how many accessions survived and produced seeds. So this is lifetime fitness, the product of survival and seeds, and either under drought and rain, in either Madrid or Tübingen. And so what you see right away, that low density is worse than high density. If you grow lots of plants together, they do on average better than if you grow them by themselves. Madrid is a harsher environment than Tübingen, so you see in Madrid, especially here, low density, many accessions die, never produce seeds when you don't provide sufficient amount of water, whereas here in Tübingen, even if you limit water, quite a few can still survive and produce seeds. And then if you provide water, you see that they can provide more, that they can produce more seeds. But you also see here in Madrid, if you give the same amount of water as in Tübingen, because it's overall warmer, the plants produce more seeds than they would in Tübingen. So Moy then did something really very similar. We knew the genome sequences of all these accessions, so when he went along the genome, and in every place of the genome, he asked if you had one allele versus another allele, and Arabidopsis thaliana is selfing and therefore homozygous everywhere, asked whether having one allele versus another allele, whether that made a difference in lifetime fitness. And so having this information, then he could ask whether the place where an allele is most likely to be found, whether that is correlated at all with the advantage it provides, either directly or through other alleles to which it is genetically linked, provides an advantage or a disadvantage in a different place. So what we're looking at here is selection in Madrid. Green is good, positive selection. Red is bad, selection against. And the alleles, so how we do this is we map out where alleles are found in which accessions across the distribution of the species, draw an envelope around it, take the centroid, and we take that as the place where the allele comes from. So we look at this by precipitation seasonality, so it's the coefficient of variation. If it's high, then that means that precipitation is more uneven. If it's low, it's more even, and then the total annual precipitation. And so here we're looking at what happens in Madrid, and it's quite clear alleles that come from places that they're more similar to Madrid, especially in terms of precipitation, but also in seasonality here, they on average do much better than alleles that come from places that are more like, for example, Tübingen with more precipitation and lower seasonality. So home alleles on average are much better. You might say, what's the big deal here? We sort of knew this already. This is partially true. Reciprocal transplantation experiments, of course, have a very long history in ecology, but you normally only know at the level of an entire accessions whether it will do better in one place or the other. So here it is really at the level of the alleles in every allele in the genome. And this is here an integrated measure because we are not only looking at causal alleles, which are much smaller in number, but really all the alleles, again, also their effects that are indirect because they are linked to causal alleles. So having this information, then, for example, we can ask what will happen in the future, and we'll see that there will be a northward wave of selection. So this is selection intensity today. You see that it is quite high around the Mediterranean, which makes sense because there is less precipitation, and you'll have more, you have higher selection, in particular here in Central Europe. You see that selection intensity actually doesn't change all that much around the Mediterranean. So what we then see is that plants have to adapt to a changing climate, and the important question is how quickly plants can adapt. And again, experiment that Moy carried out when he was a doctoral researcher in the lab, a different type of experiment, this time just in the greenhouse, where he again used hundreds of accessions from the 1001 Genome Project, and they did a very simple experiment. He watered them for a few weeks, and then he withheld water, and there was a little accident here. Somebody saw the plants, they looked really poor, and re-watered them. That wasn't really supposed to happen, but you see that overall at the end of the experiment, very little water left. And then he imaged these, and here we're just looking at green pixels, and you see initially how the plants nicely go, and then the green pixels disappear. So it's not that the plants get eaten here, it's just the green pixels disappear because the plants dry out, and you see that different plants happens to a different extent. So for every plant, you can then count the green pixels and plot them. And so here this decay in green pixels can be fit to a quadratic equation, and the quadratic parameter presents a nice proxy for a drought survival index. And so here's an example of a plant that is doing well, that towards the end of the experiment is still quite green, and here's a plant that does not do well, that towards the end of the experiment does not look very good anymore. So we can then project this back on the map. We know where accessions come from. We conduct environmental niche modeling using random forest, and then can predict for the entire map, not just the places where we've sampled actually Arabidopsis thaliana, how likely it is that plants will survive under the local environment. So again, we can ask how are good and bad alleles distributed. I already alluded to this. So we perform GWAS. We get somewhat over 100 alleles that are probably causal for survival under drought. And then for each of these alleles, we can map out where they are on the landscape. And there we, of course, use not only the 211 accessions in the experiment, but all the accessions from the 1001 Genomes Project. So we get a map of the distribution of individual alleles and individual genes, and then we can stack all the maps, and we get a density map of alleles that are favorable under drought conditions. And so here, examples of exceptions from Sweden, from Germany, and here Madrid, how they do under drought survival. And you see here that particular center of Europe, there are relatively few alleles that support survival under drought. So having this information, what the distribution of alleles is today, we can ask how many more alleles do accessions need to survive in future, say in the year 2070, and we can compare that with the actual number of alleles that are present, and that gives us an adaptation deficit. So how many alleles will be needed in future versus the alleles that are present there today. And so what you see again here around Central Europe is actually the most challenging environment. Plants will need more alleles than are actually available in these local populations. That is not necessarily what one, I think, automatically would have thought. That again, here around the Mediterranean, plants will actually do relatively well because there is already intense selection and they're quite well adapted to drought conditions. So if there are places where the genetic material is not present basically to adapt, that even if you could combine all the alleles that help the plant to adapt to drought, if you could combine all of them in a single genotype, then obviously the only way to adapt is, again, either by migration or perhaps by new mutations. So that's what I'm going to spend the rest of my talk about. So I think you all know that there are many sources of mutations: replication errors, spontaneous deamination, reactive oxygen species, UV, and so on and so forth. And that happens in all in all species. Now, fortunately for us and fortunately for the plant, most DNA damage is repaired. So there's direct reversal of damage, single-strand damage repair, double-strand damage repair. And these mechanisms are really very, very, very effective, but of course they are not perfect. And so the damage that is not repaired, that remains, those are the mutations that then are transmitted to the next generation, either the next generation of a cell or the next generation of individuals. Now, a central tenet of Darwinism, of neo-Darwinism, is that mutations are random with respect to their outcome. Mutations are, of course, not random along a chromosome. That has been long known that certain features, such as GC density, tend to affect mutation probability, but again, the central idea is really that the mutations are random with respect to the fitness outcome. And it is the sieve of natural selection that then removes certain mutations. So these are the initial mutations that occur. We have the sieve of natural selection, and then what we observe in a population is the result of mutational input and then selection, and certain mutations would be removed. Now, how can we measure the natural mutation spectrum and rate? And so a great tool for this are so-called mutation accumulation lines. Very simple idea. You have an ancestral line which you split up, and then you propagate these sublines for a certain number of generations, and then at the end you ask what mutations do I find in these MA or mutation accumulation lines versus the ancestral line. And this is from a review by Halligan and Keatley, and should also point out, if you work with a selfing organisms such as Arabidopsis thayana, half of the mutations would be lost upon selfing because the wild-type allele would be fixed, and other half would become fixed, so as in this example here. Now, Mutation accumulation lines have actually a very long history, almost 100 years, and that history goes back much, much longer than our ability to sequence genes or even genomes. And so Muller, who later got a Nobel Prize for his work on mutations, was thinking about mutation. Initially, he was almost despairing. So he thought that we know that mutations must exist, but maybe it's something that we will never be able to directly observe. Actually, in this paper in 1928, he presented a nice way to observe mutation. He did introduce mutation accumulation lines in Drosophila in this paper here. So we were very fortunate that Ruth Shaw had generated mutation accumulation lines for Arabidopsis thaliana, and that she had done so in the Columbia 0 reference background. So when it became possible to sequence entire genomes with short-read sequencing, this is an early Illumina sequencer, one of the first things we did was to get these lines from Ruth and sequence them and get an overview of distribution of mutations across the 5 chromosomes of Arabidopsis thaliana. And so we measured the mutation rate of about 7 times 10 to the minus 9 per base pair and generation. It's about 1 mutation per haploid genome and generation, or about 0.2 non-synonymous mutations per haploid genome and generation. And this is really quite interesting, these 0.2 non-synonymous mutations per haploid genome and generation in 10 generations, so that would be about 2 such non-synonymous mutations. And independent work on mutation accumulation line by John Willis had estimated that every 10 generations there would be 1 new deleterious mutation arising that was based entirely on phenotypic analyses. Now, this work was done in the lab, and you could say, you know, maybe in nature it's really very different. So at that point, we could have taken some seeds and thrown them out in nature, come back 10 years later and ask how much, how many mutations do you see, and that's certainly something that we considered. But at the same time, we had been genotyping a lot of lines in preparation for the 1001 Genomes Project. And in Europe and Asia, what we had seen is that you basically never see the same genotype more than once, except when you are at the very same place. A very different picture in North America, where it turned out that the same multi-locus haplotype could be quite widely distributed. And specifically, there was one genotype, which we called haplogroup 1, because it's such a common genotype, that accounted for almost half of the lines that we had from Arabidopsis thaliana. This was just based on 149 markers, but these 149 markers identical in accessions from the East Coast all the way to the West Coast. So what we hypothesized that this was maybe a natural mutation accumulation line experiment. Of course, selection would have been acting on these lines, but that there was a line that was introduced to North America relatively early on by Europeans and then spread all over North America. And in principle, that should allow us to measure mutation rate, because say if it arrived 400 years ago or so, and then we have modern lines that were collected at slightly different times, let's say 1990 and 2010, there would be a difference in time here. So this line here would have more time to accumulate mutations than this one here. Of course, this difference in time, about 20 years, is relatively small compared to the total age of this population, if it is indeed about 400 years. So what would be much better is if we were able to sample lines from somewhere back in time, because then they should differ much more by the number of mutations that they have. Now, an optimal method to do this would be a time machine, but we did not have access to such a time machine. But it turns out there are actually time machines, and I really have to credit my postdoctoral advisor, Elliot Meirowitz, for pointing this out to me about 10 years ago. And these time machines are herbaria. Herbarium samples of Arabidopsis thayana are plentiful. And I'm sure you all know that it's possible to sequence ancient DNA that is tens or even hundreds of thousands of years old. So Elliot suggested that we see whether we can sequence herbarium DNA from herbarium samples. And you have to take certain precautions to make sure that you don't contaminate it with modern DNA. And so this is work that was led by Claude Becker in close collaboration with Anand Bobano, who is a specialist in ancient DNA. So we assembled a collection of herbarium specimens going back all the way to the 1860s. You can see that every decade, basically, we had some samples, and then we had these modern samples here as well. And so we found something like 5,000 mutations in the samples. Blue again, herbarium. You can see in this phylogenetic network. This is a network. You don't see any reticulations because it actually behaves like a clone, as we had hoped. You see that on average the branches for the herbarium samples are shorter because they had less time to evolve. And then we can just take the time at which any of these samples were collected and compare pairs and plot the number of mutations that distinguish a specific pair against the number of years between samples having been collected, and then you can triangulate the mutation rate. And so we come up with something like 2 to 5 times 10 to the 9 per base per year, which is on the same order what we had found for the mutation rate per generation in these lab lines. These plants grew outside, and we of course don't know how many generations there are per year, but it will be something between 2 generations per year to 1 generation every other year. Now, having these data also from more lab-grown mutation accumulation line, we asked whether there perhaps is something else interesting in the mutation accumulation line data. And so what we had noticed a number of years ago already, that there seemed to be more mutations near the centromere than on the chromosome arms. And we didn't quite know what to make from this. But looking at this pattern here, it is of course reminiscent of patterns in epigenetic marks. Here is chromatin accessibility, here is CG methylation, and looking along the 5 chromosomes of Arabidopsis thayana, see, for example, that CG methylation is much higher near the centromere than on the chromosome arms, and conversely, chromatin accessibility is much lower near the centromeres. So Gray Monroe had the great idea to think about whether there might be a connection with epigenetic marks and mutations. Now, before doing this, we wanted to reassure us that there really was very little selection in the mutation accumulation lines. If you look at the ratio of stop to synonymous mutation or non-synonymous to synonymous mutation, you see here compared to a null model where we assume that the mutations are randomly distributed over the genome, given, of course, sequence characteristics and so on, see that the mutation accumulation lines are very close to the null model, both the ratio of stop to synonymous and non-synonymous to synonymous mutations, whereas what we observe in wild lines in the 1001 Genomes Project is very different. So Gray recalled mutations in the mutation accumulation lines. He is now an assistant professor at UC Davis, and then teamed up with a doctoral researcher, Tanvi Shrikant, who is now a postdoc at ETH, and we also had help from the Fenster, Rutter, Wang, Clevenshine, Imbert, and Argonne labs on this. And Gray built a model using both germline mutations and then about 6,000 somatic mutations as well, and used information on gene expression, GC content, cytosine methylation, and so on to model probability that mutation would arise in different parts of a gene or outside of genes. And so for single nucleotide variants, you see, for example, that GC content is a very nice predictor. And also for insertion deletions, you see that GC content, local GC content is a very nice predictor. But then also see here various chromatin modifications are good predictors. And the model is being built in a way that redundant variables are excluded as much as possible. So having now a model for what features in the genome lead to mutations, we can then walk along the entire genome and annotate the entire genome for mutation probability. And I should mention that he got very similar results when he excluded coding regions for training. So here we're looking not at observed mutations, but mutation probability as predicted by our model. You see here that around transcription start site, that mutation probability much higher in front of transcription start sites than downstream of them. So this would be outside of genes. This is in genes. This is go down again is due to the fact that you'll, as you move away from a gene, the chance of being in a gene again goes up. And similarly here, right downstream of the transcription start site, you're in a gene, but as you move away, then of course the chance increases that you at some point move outside a gene again. And then the opposite pattern here around transcription termination site, in front of the transcription termination site, inside a gene, low mutation probability, and then it rises dramatically downstream of the transcription termination site. Does this agree with what we observed? This is the germline mutations, mutation accumulation lines, and you see a pattern that's very reminiscent of this. Somatic mutations in the same lines, somatic mutations in a different paper, and then these were the observed polymorphisms in this North American population. Turns out that polymorphisms in 1001 genomes are also very similar, so many more polymorphisms outside genes than inside genes. And the density of polymorphisms and how they are distributed across different accessions is captured by different metrics, especially by Tajima-SD. You see Tajima-SD that it is much less negative inside genes, which is compatible with the idea that mutations are less frequent and also more rare in a population inside genes than outside genes. Gray also looked along each gene with this mutation probability score, and so broke it down to the genes into all the genes that have 3 exons, 4 exons, 5 exons, and so on and so forth. And you see that the mutation probability always highest in the first and in the last exon, and lower mutation probability in the interior exons. And that's also what we see in the 1001 genomes data, and then this mirror image pattern for Tajima's D, this population genetic metric. And this is not just restricted to Arabidopsis thaliana, it's a similar polymorphism pattern in Populus trichocarpa, published data set from 544 accession. So the final thing then that Gray did was to ask, when he computes mutation probability for individual genes, is there a difference between different gene classes? So here you see the scaled mutation probability score for all genes. You see it's quite a range here. The bottom decile is this blue here, this pigeon blue, and then the top decile is these ochre here. So he asked whether genes that are likely to mutate, are they different from genes that are less likely to mutate? And so he again drew on the published data. So for example, lethal versus non-lethal mutant phenotypes. So these are mutations in genes that are required for survival. These genes you can mutate and the organism can still survive. You see that the mutation probability is lower for, and this again, this is not, in this case he has not observed mutation, but mutation probability lower for these lethal effect genes than the non-lethal effect genes. Similarly here, essential genes, or those that have a morphological phenotype, biochemical or conditional phenotype, see that it's lowest for essential genes. And this does agree with direct observation. You have to take this a little bit with some grain of salt here, because we're looking at somatic and germline mutation. And somatic mutations, they would be mostly heterozygous, and therefore we should be able to observe mutations in genes that are essential, but of course they cannot be transmitted to the next generation when they affect gene function. Although again, proviso, most or many mutations in the gene, of course, do not affect gene function. So obviously there are mutations in essential genes that can be transmitted to the next generation. For example, mutations in synonymous sites, or many mutations in introns. So observed mutations you see here is lower encoding sequences for essential genes than genes that have conditional phenotypes. And you also see this in introns, and obviously in introns many fewer mutations would affect gene function. So here you can argue that at least some of the deficit that we see is due to a substantial number of mutations affecting gene function, therefore not observable. But in introns, of course, that number should be much smaller, and we still see a very similar difference. And then when we look finally for GO enrichment, we see indeed that there are very different genes in bottom and top mutation deciles. So here, the bottom decile of mutation probability, these genes are annotated with in categories such as translation and splicing, so processes that are required in every cell, whereas the genes in the top mutation decile, they are much more likely to have functions in response and regulation, so have conditional roles. So this, of course, is rather spectacular. You might say, well, fine, I find this a little bit difficult to believe unless you can present a plausible mechanism for why there should be a difference in mutation rate in, say, non-essential or essential genes. And so, of course, I already told you that the model was built using epigenetic information. And if it is built epigenetic information, then you'd expect that epigenetic factors would be correlated with these mutation probability. So here again, we are looking at observed de novo coding sequence mutations, both somatic and germline, and this is plotted against, for example, the density of synonymous polymorphism per base pair, or this is divergence between Arabidopsis thaliana and sister species Arabidopsis lyrata, or here the ratio of non-synonymous to synonymous polymorphism. Here also variance in environmental variance in gene expression and genetic variance in gene expression. And this perhaps particularly interesting, so you see that genes that are more variable in expression, they are more likely to have coding sequence mutations than genes that are less variable in their expression. And so here is one such epigenetic mark that shows a very nice mirror image pattern, H3K4 monomethylation density per base pair. And you see that it is when it is very low, then we observe a large number of synonymous polymorphism. If it's very high, we observe a low number of synonymous polymorphism. Here, when the mark is high, when the mark is low over here, we have a few polymorphisms. So what is the bottom line then? How do we think about this? So at the level of individual genes, we think that gene bodies have different epigenetic marks than outside genes. That, of course, is not controversial at all. And then that these different epigenetic marks differentially attract factors for DNA protection and DNA repair. And there's plenty of evidence in the literature that connects epigenetic changes to DNA repair. This is just one paper here from a few years ago. And so similarly, then what we propose is that housekeeping genes as a class have different epigenetic marks than non-essential genes, for example, those that are induced depending on the environment, and that they differentially attract DNA protection and repair mechanism. And this is really our main discovery then, that we found that essential genes have, on average, as a class, different epigenetic marks than non-essential genes. And connecting this to what is known about this relationship between epigenetic modification and DNA repair can serve as a basis for these genes being more protected from DNA damage or more efficiently repaired after DNA damage than these genes here. And again, this is perhaps the most provocative study we ever published, as I announced in this tweet a number of years ago. Now, all this work on mutation has really made me think very differently about the natural world around us. This is a wheat field here, a very large wheat field, and I think most of us who would look at this field, so our first reaction would be, well, these plants are all identical. They are all from the same variety of wheat. But this mutation rate, about 10 to the minus 8 per base per generation, will not be very different in wheat. And so when you then think about wheat genome, 20 gigabases, it means there should be about 200 mutations in every grain of wheat that you see on this picture here. Now, a hectare in a good place and in a good year can produce up to 10 tons of wheat. So this would be 200 million grains of wheat per hectare. If we put this in here, it means that we have 40 billion mutations in the harvest from a single hectare field, or about 2 mutations on average for every place in the genome. So these mutants are all different, and even, or these plants are all mutants, and they're all different from each other. And even on a 1 hectare field, which is much smaller than the field that you see here, you would have mutations in about every place in the genome. And so what I want to then finish with is that natural mutations are really ubiquitous, and what we're doing with genome editing, which really is nothing else than target immunogenesis, is not very different from what happens in nature anywhere. It's just much easier to find the plants that have a mutation at the specific place that we are interested in. And here I think it's very obvious why Nobel Prize were given for the discovery that the CRISPR-Cas system could be enlisted to make genome editing much more efficient than it was before. And I would like to point out that even the Intergovernmental Panel on Climate Change, the IPCC, points out that one should consider genome editing as a tool in plants, and I really advocate that we make use of all the tools that we have to breed plants that can better deal with the climate change that we're all facing. And with that, I'd like to close. This is a picture from last year, as you can see in the middle of the pandemic, a farewell picture from the roof of our institute overlooking the Swabian Alp. And with that, I'll stop sharing and see, and I'm happy to answer questions.
If there are any questions, people, you can just write in the comments. I mean, even if you write in Farsi,
if there are any questions, people, you can just write in the comments. I mean, even if you write in Farsi, I can just translate it. It will be fine.
There are 2 people Okay, first Nassim Alijani.
I think there are two people. Okay, first Nassim Alijani. As we can actually what happened.
Okay, first yeah. I have to activate the microphone.
So I think that there are a few people who would like to ask questions, but their microphones are muted. So.
Activate the microphone.
Yeah.
So I have
activated question
for Rasuli.
Yes.
Yes.
So I think you can ask your question.
First of all, Professor Weigel, thank you very much for this excellent and perfect lecture that you are
Professor Weigel, thank you very much for this excellent and perfect lecture that you are.
And I enjoyed to participate in the lecture from this time of moment. Really, I have a question about one of your slides that you show a chemical structure of a toxin. If I were true, it was a chemical structure of aflatoxin B1. I found that this toxin and some issue relevant to this toxin may trigger the rate of mutation in some plant. Just recently, I have done research on toxin, and I found that the presence of some plant pathogen in the nearby of crop rhizosphere can trigger some mutation in plants and may affect their productivity. So the question that I have is that how fungal toxin, in the case of aflatoxin, though they are highly toxic for human and animal feed, can change the rate of mutation in plant. Is there any possibility such toxin produced by exotic microorganism or symbiotic microorganism affect the mutation and epigenetic issues in plants, especially in crop plant that presently we have a great problem with controlling of plant pathogen and insect in the production sites? Thank you. If you can give me some clue or recommendation that help me to discuss this issue in some research that we have done. I can't hear anything at this moment. Excuse me, can you hear me at this moment? Excuse me, I lost your voice. Yeah, no, I can hear you. Did you hear all of my question?
For survival. What happened to
Dr. Kalirat, can you hear me?
Yes, I can hear you. I just can't hear Professor Weigel. Why is he mute? Why?
Now it should be fair. I think it was when we moved out. So yeah. So sorry. Yeah. So I said that given what we know about fungal toxins causing mutations in humans, I wouldn't be very surprised if fungal toxins also increase mutation rate in plants. That makes perfect sense to me.
Yes, Professor.
Professor
Professor
Weigel,
Weigel,
my
my
explanation was
explanation
that
was that
how fungal
how fungal
toxin
toxin
can affect
can
the
affect the
occurrence of
occurrence
mutation
of
in
mutation
plants.
in plants.
I recently
I
have done some
recently have
NGS
done some
analysis,
NGS
and I
analysis,
found
and
that this
I found
toxin
that
in
this
plants
toxin in plants
have the ability
have
to
the ability
interact with
to
some
interact with
protein
some
that
protein
affect the
that
productivity
affect
of
the
plants.
productivity of plants.
Can we assume
Can
that
we
such plants
assume that such
during
plants
this upcoming
during this
climate change
upcoming
affect
climate
the
change
quality of crop
affect the quality of crop
productivity by affecting
productivity
their
by
epigenetic
affecting
issues
their
and
epigenetic
something
issues
like this?
and something like this?
Can you please repeat that again? So you're saying is what contribution could epigenetic change make to adaptation to climate change?
Excuse me,
Excuse me,
I have a bad voice from the meet.
I
I don't
have a
know what
bad
happened,
voice
but
from the
I
meet.
repeat my
I don't know
question.
what happened, but I repeat my question.
In one of your slides, you have shown the
In one
chemical
of your
structure of
slides,
an
you have shown the
aflatoxin.
chemical
If
structure of an
I were
aflatoxin.
true, it was
If
aflatoxin
I
B1
were true,
that
it was
produced by some
aflatoxin B1
Aspergillus
that
species.
produced
So
by some
this toxin cause a significant mutation rate in human body and animal. But recently, from some analysis that I have done, I found that this toxin also have the ability to change the ratio of mutation occur in plant, especially in crop plant that dynamically interacted with a fungal toxin and fungal species. So can we assume such toxin be considered as one of the environmental factor to change the epigenetic mutation rate in plants or in the crop productivity?
I actually don't know. Sorry.
Okay, thanks.
Okay, thanks.
So there was actually a question in the chat. From Ali Jamali. So if we can use mutants to control climate change.
Yeah, so I think where that question goes to is there an optimal amount of mutation rate? So I think that is a big question in evolution. So with a very low mutation rate, it is obviously more difficult to adapt with higher mutation rate. But on the other hand, with a higher mutation rate, it's also more likely that you have mutations that are detrimental. It will be super interesting to simulate what is actually the optimal mutation rate for adapting to a certain speed of climate change. We haven't done that, but I think it will be really interesting to do.
So any more questions? By the way, whenever someone is talking, I need to mute you to avoid reverberation. Hopefully it's fine. Right, was there any more questions? Is there someone? Okay, Nassim Alijani. So do you need to speak or do you want to type your question? Okay, so just ask a request, raise your hand and I'll just allow you to speak. Perfect, go ahead. Nassim Alijani, you have microphone so you can ask your question. I'm sorry. The microphone was just touch your question. Okay. We lost that question for whatever reason. So if someone has a question, please raise your hand. So actually, I have a question. In the meantime, so there was actually I think if you followed the Twitter, there was like a minor meltdown with regards to the paper on the nature of mutations. I was wondering what is your perspective on this specific question of the randomness of mutation, because as an evolutionary biologist I always assume that mutations are just merely indifferent towards the consequence or the fitness consequence. So what do you think about the reaction there, and what is your own interpretation with regards to the result and what is generally described as New Darwinian framework?
Yeah, I mean, we were certainly very much surprised. We didn't start with the hypothesis that there would be this difference in mutation rate, but this came from noticing after plotting the patterns of mutations. This came after we then saw that there was this relationship with epigenetic features. And then going into the literature and seeing that actually in cancer biology that there is plenty of support for the idea of epigenetic features being related to mutation rate. So I think if 3 years ago or so somebody else would have published such a paper, I would have reacted extremely allergically as well. Of course, we were extremely worried that there is some artifact in the data in our analysis, and it's still a worry that I have that we overlooked something trivial in the data, or that we overlooked some technical artifact. We tried really hard to think about what errors we might have overlooked. I'm still not 100% sure that we have overlooked all possible errors, but as I said, we did a lot of experiments and different types of analysis that seem to indicate to us that the observed trends were real and not a technical artifact. If somebody else finds out that in the end this is due to the data being problematic, I would be the first one to accept that, because my old view would be restored.
Perfect. So if there are more questions, I think we probably can handle one. I think if there are no more questions, oh, actually, there's another Johnny. Please go ahead. Just go ahead with the question. I open the microphone for two participants who want to ask questions. Okay, there seems to be a question. So if you have more questions, Professor Weigel is available online through Twitter, and I think would be more than happy to answer your questions. Again, it was a pleasure to introduce Professor Weigel, and also it was a fantastic talk about a very interesting topic, which was real. Again, I would like to thank the Student Scientific Association of Agronomy and Plant Breeding at the University of Tehran for organizing something like this, and I think it is fantastic finding and very interesting discussions about the nature of mutations.
Thank you.
Thank you so much. Okay, thanks everyone as well. Hopefully you found a lot to think about after this talk.
Thank you again, everybody.
And thanks again for Professor Weigel for giving this fantastic talk.
Thank you.